Welcome to the debate. Imagine going to the doctor for severe depression, or maybe to find out if your cancer treatment is going to work. And instead of drawing your blood or doing a brain scan, they analyze your stool.
Yeah, it sounds like pure science fiction.
It really does. But the microscopic ecosystem living inside your digestive tract might actually be a more powerful diagnostic tool than your own DNA.
I mean, it is a staggering concept, but also it's the absolute definition of diagnostic complexity. We are used to clinical tests being highly controlled and singular, like a standard blood draw measures one specific protein.
Right, it's a closed system.
Exactly, it is a closed system. But when you look at the human microbiome, you are looking at 100 trillion living microorganisms constantly reacting to their environment, to each other, and, to the host. We are moving from looking at a single tree to trying to read the health of the entire forest based entirely on the shifting soil.
And whether we are actually ready to do that is exactly what we are dissecting today. We are looking at the clinical translation of microbiome-based diagnostics.
Which is a massive undertaking.
Oh, absolutely. Drawing directly from the latest academic symposium literature and multi-omic research, we have to ask, are these microbial signatures, the ones found in the gut, the lungs, and localized tissues, are they robust, independent markers that are ready for precision medicine?
Or conversely, are they currently too confounded by human variants and bidirectional disease dynamics to serve as standalone clinical tools?
Right. And I'll be representing the position that, yes, advanced multi-omics and machine learning have successfully distilled actionable, independent diagnostic signatures from the microbiome, and they are ready to revolutionize clinical practice today.
And I will be taking the opposing view. I represent the position that profound, underestimated human variants, methodological inconsistencies, and the bidirectional nature of disease make these microbial profiles supplementary at best in their current state.
So let's just dive into the position statements. Historically, sure, the microbiome was treated as a black box. Interesting associations, but no real clinical utility.
Yeah, lots of noise.
Exactly. A lot of noise. But today, the analytical tools have finally caught up. We can confidently say the microbiome is a measurable, causal driver of human health. By applying machine learning to shotgun metagenomics, which is essentially reading all the genetic material in a sample at once, we are seeing highly accurate diagnostic models.
But how accurate is highly accurate in a clinical setting?
I mean, incredibly accurate. For example, relying solely on taxonomic profiles, algorithms can now classify subjects with major depressive disorder, MDD, with a balanced accuracy of 0.90.
Oh, wow. That is a high number.
Right? And it means we are beginning to outperform traditional tests. Take the Biome 1 stool test. It predicts a patient's response to immune checkpoint inhibitors with greater sensitivity than standard PD-L1 expression tests.
Okay, wait, so a stool test is beating a direct tumor biopsy?
Exactly. It gives us a more accurate picture of a patient's immune priming than their own cellular markers. These models aren't the future. They are here right now.
Well, I have to approach this with a stance of intellectual caution. The landscape of clinical microbiome research is undeniably fascinating, I'll give you that. But it is currently plagued by vastly underestimated biological and methodological variance.
You think the variance is still too high?
Oh, definitely. Look at where the foundational data comes from. The vast majority of human microbiome data derives from individuals living in socioeconomically developed nations. This creates a massively skewed interpretation of what constitutes a normal or diseased microbiome.
Okay, so a demographic bias?
Exactly. It completely ignores critical factors like distinct ethnicities, geographical environments, and the natural physiological shifts that happen as we age. Because the relationship between the host and the microbiome is inherently bidirectional, what we often label as a diagnostic signature might just be a secondary reflection of host inflammation or environmental exposures. It's not a reliable, independent marker of disease.
I hear you on the environmental noise, but that's exactly why we don't look at the whole chaotic ecosystem at once anymore. When we isolate specific, tightly knit microbial networks, that environmental noise drops away. The predictive power becomes incredibly sharp across diverse diseases.
But how isolated can a network really be in an ecosystem of 100 trillion microbes? Everything is connected.
Very isolated, actually, if you know what to look for. In dental medicine, researchers have built an 11-species diagnostic model that successfully distinguishes between varying grades of periodontitis. It specifically differentiates stage 3 grade B from the much more rapidly progressing grade C.
Just by looking at those 11 species?
Yes, by tracking the specific overgrowth of pathogens like Capnocytophaga. Or consider reproductive health. A simple swab showing the enrichment of a specific taxa, Lactobacillus crispatus, acts as a clear indicator of early pregnancy loss. These are highly specific, reproducible signatures.
Okay, you're pointing to an 11-species model for dental disease, and while that's neat, I'm sorry, but I just don't buy that this scales to systemic diseases. Let me tell you why.
Go ahead.
The idea that we can isolate a specific microbial network and call it an independent diagnostic marker just ignores the fundamental reality of how microbes interact with their environment. Let's look at the recent research on tuberculosis.
TB is a great example of a systemic infection. What did the signatures show?
Well, researchers tried to find a definitive gut or lung microbial signature for TB susceptibility and progression. And they found signatures, sure, but those signatures were almost overwhelmingly confounded by external variables.
Such as?
Diet, recent antibiotic exposure, geography. These factors completely altered the microbial landscape. The microbiome isn't a static blueprint. It's more like, the economy of a bustling city.
Meaning what, exactly, in a clinical context?
Meaning if a factory shuts down, say a sudden dietary shift, or a natural disaster hits, like a course of broad-spectrum antibiotics, every single neighborhood in that city reacts. The economy halts. So how can a clinician trust a standalone microbial diagnostic signature for a systemic disease if a patient's recent course of antibiotics completely rewrites the diagnostic readout?
I see why you think that. But let me give you a different perspective. You are characterizing the microbiome merely as a reactive economy, just absorbing shocks from the environment. But the science has moved beyond these basic compositional surveys.
Oh, how so?
We are now using functional metagenomics, which proves direct mechanistic causality. Basic metagenomics tells us who is there, right?
Yeah.
But functional metagenomics tells us what they are doing. We now know these microbes are actively driving the disease state.
Okay, explain the distinction there. Give me an example.
Let's go back to major depressive disorder. It isn't just that depressed patients happen to have a different microbial composition. Functional analysis shows that the specific depletion of a gut bacteria called Faecalibacterium prosnitzi directly correlates with a decrease in cuisine synthesis.
And for the sake of clarity, why is cuisine synthesis critical for a psychiatric condition?
Because your body needs cuisine for essential transfer RNA functions. And those synthesis pathways are directly linked to the availability of monoaminergic neurotransmitters. We are talking about the exact chemicals, serotonin and dopamine, that are depleted in clinical depression.
Wait, so the bacteria is literally involved in making the neurotransmitter precursors?
Yes. When that specific bacteria dies off, the chemical assembly line in your gut stops, the brain doesn't get the precursors it needs, and the patient experiences clinical depression. The bacteria isn't just a marker, it's actively driving the disease. And we see this in metabolic dysfunction-associated fatty liver disease, or MAFLD, too.
With the altered bile acid signaling, right?
Exactly. It's the actual bacterial translocation and altered bile acid signaling that actively drives lipotoxicity and fibrogenesis in the liver. We are mapping the exact biochemical pathways where the microbe causes the pathology.
Admitting that we can map a metabolic pathway from a gut microbe to a neurotransmitter is a profound scientific achievement. I will grant you that. But finding altered metabolites absolutely does not solve the bidirectional chicken or egg problem for diagnostics. Let's examine inflammatory bowel disease, or IBD.
Okay, IBD is deeply tied to the microbiome, yes?
But look at how it's tied. In IBD, a combination of the host's genetic risk and environmental triggers causes profound inflammation in the gut tissue itself. This host-driven inflammation fundamentally alters the local microenvironment. When the gut is inflamed, blood flow increases and oxygen leaks into the gut lumen.
Which is a huge problem because most of our beneficial gut bacteria are obligate anaerobes. They literally cannot survive if oxygen is present.
Exactly. The host's inflammation introduces oxygen, which acts like a poison to the healthy microbiome. It wipes out the beneficial microbes and preferentially selects for facultative anaerobes, the weeds of the gut that thrive in that toxic environment.
So you are arguing that the host's physical state creates the dysbiosis, not the other way around.
Precisely. And the same logic applies to the mental health example you just gave. Host psychological stress induces neuroinflammation, which sends signals down the gut-brain axis, altering gut motility. The host changes the environment first.
I see.
So if host inflammation fundamentally alters the gut ecosystem, aren't we just diagnosing the host's immune response in a highly convoluted, expensive way, rather than identifying a primary microbial defect?
That is an interesting point, though I would frame the clinical utility differently. Even if the host initiates the inflammatory cascade, the resulting microbial dysbiosis becomes a self-perpetuating engine of the disease.
A secondary engine, though.
Secondary or not, once those facultative anaerobes take over, they produce metabolites that cause even more inflammation. Breaking that cycle is critical, which makes measuring it clinically vital, regardless of who started the fire.
But can we reliably measure that cycle amidst all the noise? That remains my core concern with all of this.
We can, because technological advances are solving these methodological issues right now. We're no longer relying on basic 16S sequencing, which is basically like looking at a blurry license plate and only being able to tell it's a Ford. Now we are using multi-omics. We combine metagenomics, metatranscriptomics, and metabolomics.
So we are looking at who is there, what genetic instructions they are actively reading, and what chemicals they are producing.
Exactly. It maps precise active pathways in real time. Take periimplantitis, a severe inflammatory condition around dental implants. By using metatranscriptomics, we don't just see a shift in bacteria, we see massive spikes in galactose metabolism and proteolysis, the breakdown of proteins. We are watching the bacteria eat the tissue in real time.
Okay, multi-omics is powerful, but it's also incredibly expensive and data-heavy. How does that translate to a routine clinical diagnostic?
Through sophisticated AI tools, there is a new machine learning algorithm called Taxacal. It takes that cheap, cost-effective 16S amplicon data, the blurry license plate, and calibrates it to match the accuracy of expensive whole genome sequencing.
Oh, really?
Yeah, it seamlessly bridges platforms, correcting the biases and giving a species-level accuracy without the prohibitive cost. AI is standardizing the field.
That's a compelling argument. But have you considered that deploying sophisticated AI on fundamentally flawed or heterogeneous data might just amplify our blind spots?
What exactly is flawed about the underlying data if the AI is calibrating it, though?
Well, you mentioned standardizing the field, but if we look at comprehensive reviews of microbiome research in colorectal cancer and chronic kidney disease, a glaring issue persists across the globe. There is massive variation in methodology and non-standard data management practices.
You mean how the samples are handled before they even reach the sequencer?
Yes, we have studies using entirely different DNA extraction protocols. Some clinics store samples at different freezing temperatures. They use different bioinformatic pipelines. When you train a powerful AI model on heterogeneous biased datasets, it doesn't magically uncover a universal biological truth.
It learns the patterns of the data it is given.
Right. It often just learns the specific biases of those cohorts. The algorithm might find a microbial signature that perfectly predicts colorectal cancer in a specific urban population in North America, but that signature might be entirely useless or even dangerously misleading when applied to a rural population with a completely different baseline diet.
I mean,
Until the underlying physical data and clinical protocols are standardized, relying on AI to patch the gaps is a massive diagnostic risk.
I'm not convinced by that line of reasoning, because it lets the perfect be the enemy of the good. Yes, biological variation exists, and methodological standardization is an ongoing process. Every field of medicine goes through this. But the clinical results we are already seeing are just too powerful to ignore.
Even with the demographic and dietary noise?
Yes, because the algorithms are becoming sophisticated enough to parse through that regional or dietary noise to find the conserved functional pathways that truly matter across all humans.
That's a huge promise for an algorithm.
But it's delivering. The fact that a microbiome signature from a stool test can predict whether a patient's tumor will shrink in response to immune checkpoint inhibitors in scenarios where traditional genomic markers fail proves that the signal cuts through the noise.
It proves there is a signal. I am not denying that.
But it proves we are moving from a reactive medical model to a proactive precision medicine model right now. The integration of multi-omics and machine learning has proven that microbiome signatures are fundamentally transforming precision medicine into a reality today. From predicting cancer immunotherapy success to identifying neurotransmitter deficits, this is happening.
Look, the mechanistic discoveries are indeed profound. Uncovering how a specific microbe synthesizes a precursor to dopamine has revolutionized our understanding of human biology. But clinical application demands a threshold of reliability and universality that I argue we simply have not yet met.
So you believe the risk of misinterpretation is still too high?
I do. The clinical application of these tools must be tempered with rigorous caution. Until we can fully account for the massive variance introduced by aging, distinct ethnic backgrounds, and geographic dietary shifts, we cannot rely on them blindly.
Fair enough.
Furthermore, until we can untangle the bidirectional relationship of dysbiosis, knowing for certain whether we are measuring the cause of a disease or merely diagnosing the host's inflamed shadow microbiome, diagnostics remain a highly promising supplement. They are not a standalone solution. The potential for misdiagnoses based on an algorithm that misunderstood a patient's recent antibiotic use is simply too great a risk.
It is absolutely clear we are navigating one of the most exciting yet complex frontiers in modern medicine. The immense complexity of the host microbiome axis, the fact that it acts like a bustling city economy, is exactly what makes it so valuable. And, it is worth noting that there is vastly more to explore in the academic literature beyond just diagnostics.
Absolutely. We haven't even touched on the therapeutic side today. The literature is exploding with interventions like fecal microbiota transplantation and the use of engineered microbes to actively correct the dysbiosis we have been debating.
Indeed, the therapeutic potential is vast. And as our understanding of these ecosystems deepens, the listener will have to weigh the emerging evidence to draw their own conclusions on just how ready these diagnostic tools are for the clinic.
It is a rapidly evolving field, and the line between correlation and causation is getting sharper every day.
It leaves us returning to that opening image. Are we ready to diagnose the health of the entire forest by analyzing the shifting, reacting ecosystem of the soil? Or are we still learning how to separate the passing weather from the true climate? Thank you for listening to the debate.